So let's talk about the phosphorylation cascade. So what is it? Well, it's exactly what it sounds like. It's a cascade of phosphorylation that is happening. And so, first of all, what is phosphorylation? Well, think back to when we talked about ATP, right? When you think about ATP, what does the ATP do? It transports a phosphate group to another molecule to make it do some sort of work. And that is what phosphorylation is, right? And so when you think about a cascade of this, well, what comes to mind? You're going to think about a bunch of molecules that are just being phosphorylated at the same time, and that's definitely 90% of the answer right there. And then the only other part of it is that it's going to be protein molecules that are actually going ahead and are being phosphorylated themselves, and then going ahead and phosphorylating other protein molecules to continue this cascade. And so let's kind of see how that works out. Well, so first, if you look right here, there is going to be a signaling molecule. And this signaling molecule is going to come from outside of the cell, and it's going to attach to this receptor. And when this happens, it's going to allow for this active relay molecule, initially inactivated, but now activated, to go ahead and turn this inactive kinase 1 into an active kinase 1. And kinase is basically the name given to proteins that basically take phosphate groups from an ATP and give it to another protein. So this active kinase 1 is now going to actually take this ATP and give a phosphate group to this inactive kinase 2. And this inactive kinase 2 is going to become an active kinase 2, and that is going to go ahead and use an ATP to give this inactive kinase 3 a phosphate molecule to turn it into an active kinase 3. And this active kinase 3 is going to give this inactive protein a phosphate group from the ATP, which is going to turn it into an active protein, and that active protein is going to do some sort of cellular response, right? And so that's basically going to be the whole point of this cascade. And now this is a hypothetical model in which there's going to be, there's only like 3 inactive kinases, but I would assume that in in actual situations, there's going to be many more kinases that are being used. And so basically, to look at like some more detailed aspects of this model, you're going to see that this ATP molecule right here can actually go ahead and become an ADP molecule. And that's basically because it's losing its phosphate. And these molecules are actually not going to stay as active kinases because of this PP right here. And PP stands for phosphatase. And this phosphatase is actually going to dephosphorylize all of these active kinases. And these kinases will return to their inactive kinase forms. And this is a very important part about phosphorylation cascade. And that is because this dephosphorylation allows for the phosphorylation cascade to happen again. And why is that? Well, that is because, again, as all of these are reverted back to their inactive forms, when another signaling molecule comes by and attaches to a receptor, this entire process can happen again.
Hi everyone, this is George. So today I'm going to talk about secondary messengers. Secondary messengers follow the path of, are involved in the path of signal transduction. And as a part of signal transduction, we can take the epinephrine signal transduction as for example. So what's going to happen first is the epinephrine messenger, also known as the ligand, is going to attach to the outside receptor. The ligand cannot enter, and once it's attached there, the G protein, which is the receptor in this case, has subunits under it. Once it attaches, the subunit, or specifically the alpha subunit, this last one, is going to move to the other side and, sorry about that, attach to the enzyme. In this case, adenyl cyclase. Yes. So after it attaches, the alpha subunit attaches to the adenyl cyclase, the enzyme will start functioning and it will take in ATP. ATP goes into adenyl cyclase, and it will convert this ATP into something called cyclic AMP, or CAMP, CAMP, represented by purple. This cyclic AMP will then a lot of cyclic AMP will be converted from ATP, and it's going to go into this protein kinase represented by these 2 green and 2 red. So this purple is going to move Anyways, okay, so it moves here, right? It moves all the way there, and then it's going to bind to the regulatory side of the protein kinase, represented by the red. The green is the cyclic part. So when it binds to the regulatory, it stops this from happening, and it allows the cyclic part to go out, and as ATP, the yellow thing, binds to the cyclic, it is now activated. And this allows for this this catalytic portion to phosphorylize, and goes to phosphorylase and basically activate phosphorylase. And what phosphorylase is going to do is it's going to go right here, and it's going to have the glycogen, and it's going to slowly break it down into individual glucose. And again, secondary messengers are involved in the inhibition of the regulatory part of the protein kinase, and the secondary receptor in our case is CAMP, again CAMP, made from ATP. So these secondary messengers, their main role is to basically produce a lot from ATP, and from this little epinephrine messenger, this becomes exponential. There becomes a lot of secondary messengers which go to a lot of protein kinases, which all go to phosphorylase and break down glycogen into glucose at a massive scale. And that's it. Thank you very much.